All right, so we mostly finished up our crash course on inheritance today in class. If we have some additional questions, we can certainly deal with them at the beginning of class on Monday. But on Monday, we're going to begin our crash course on molecular genetics. And so we'll begin with a few key terms, same way we began with our crash course on inheritance. First key term is complementary. Complementary is a description of the two strands of DNA. Really, it's a description of the base pairing. The strands of DNA are held together by hydrogen bonds, and they are held together by hydrogen bonds formed between a nitrogenous base on one strand and the complementary nitrogenous base on the other strand. We'll talk more about the complementary nature of DNA after this slide. The next key term is antiparallel. So again, there are two strands of DNA held together. The DNA strands, we read them from the 5' end, which is referring to which carbon is unbonded, and it runs all the way until the 3' end, which again is referring to which carbon on that ribose sugar is unbonded. And so we read it from the 5' end to the 3' end. One of our strands runs, let's say if you're looking at our two strands of DNA on a screen running left to right, although I think the example I have in a couple of slides runs top to bottom. But if you can picture in your mind left to right, if the bottom strand is 5' end is on the left and the 3' end is on the right, the strand on top, it is antiparallel. So the 5' end is on the right and the 3' end is on the left. Chromatin, this came up in class today, and I incorrectly used the definition of chromatin to describe chromatid, but chromatin is the chemistry of the chromosome. It is a combination of DNA along with all of the proteins that are involved in the organization of DNA. Histones. Most of the proteins that are involved in the organization of DNA into chromosomes are histones. There are 5 different types of histones specifically, and then there are other proteins that can't be confused with histones that are called histone-like proteins. But histones are proteins that DNA is wrapped around, and we'll see that in a few slides. Supercoiled. This is a description of DNA when it is put into an inactive form or even a partially inactive form. It is not only wrapped around proteins, and that would be coiled, but then the coils are coiled. The example of supercoiled is usually a telephone cord that is, tends to spiral and already begins coiled, but then those sections of it actually coil upon themselves, and you get a supercoiled cord. Heterochromatin. Heterochromatin is very condensed chromatin. It is the form of the chromosome that we see when we're looking at mitosis or meiosis, where we get that nice characteristic X shape to the chromosome where you've got your two sister chromatids held together. So heterochromatin is a seriously condensed form of chromatin. Euchromatin, on the other hand, is an uncondensed form of chromatin, and this we'll see in a few slides. This is the active form of DNA. That should read centromere. The R is missing. Centromere is sort of the middle of the chromosome. It's not always exactly in the middle, but it's near the middle of the chromosome. It's where the two sister chromatids attach together during the first portions of mitosis and during all the way up until anaphase 2 of meiosis. It's also where the microtubules attach to the chromosomes to actually move them around during those cell processes. Finally, the telomere. this is the end of the chromosome, the tip of the chromosome. So let's look at some of these in more detail. So here's complementary. Again, DNA is complementary, and what we mean by that is that there are sets of these bases, and we find 4 different nitrogenous bases occurring in DNA. From left to right, it is thymine, and then adenine, and then cytosine, and then guanine. Thymine and cytosine are pyrimidines. Adenine and guanine are purines. And then in RNA, instead of thymine, you have uracil, which is a pyrimidine found in RNA. And so these thymine will always bond with adenine because they are shaped correctly and have the correct chemistry to form hydrogen bonds. And the thymine adenine pair forms 2 hydrogen bonds. The thymine or the guanine cytosine pair forms 3 hydrogen bonds. And again, this is the complementary nature of DNA, and this is what holds the 2 strands together. It also is what we'll see on Wednesday, what actually helps DNA get replicated and what helps RNA get written from DNA. Also, as we discuss DNA, we've got to talk about antiparallel. We already defined this, and I asked you to visualize a strand running left to right. Then I remembered and even mentioned that our example runs top to bottom. But you can see right here, let me get my pen. You can see right here we've got a free carbon 5 that is not bonded to anything, and on this end we have a free carbon 3 that is not bonded to anything. These would be the carbons that would typically be bound to a phosphate group holding the 2 sugars together. You can see the phosphate group is bound to carbon 3 on this sugar and carbon 5 on the next sugar. And so the end with that free carbon 5, we call that the 5' end. The end with the free carbon 3, we call that the 3' end. And so if the strand on the left runs 5' to 3' as you move down, the other strand has to move in the opposite direction, and it is drawn accordingly, with even, helpful, to see the phosphate groups represented upside down. So DNA is both complementary and it is also antiparallel. All right, chromatin. So again, chromatin is the DNA along with the proteins that work in organizing the DNA. So over here on the left, we've got the super condensed chromosome that we see during mitosis and meiosis. It's pretty much the only time you see chromosomes structured like this. Here there are 2 sister chromatids. Here's one on the right and one on the left, and those are identical until crossing over will exchange some of the genetic information. Now, chromatin, even when the chromosome is not in its fully condensed form, the chromatin is still made up of both proteins and DNA. And so you can see over here is what's called a histone octamer, and it is made up of 2 units each of H2B, H2A, H4, and H3, and it's an octamer made up of 2 of those each. And so the quaternary structure of that histone is all 4 of those protein subunits combined together to form our overall structure, and you can see the DNA wound around this histone. And so what that does is it functions to condense the DNA a bit, but this is not right, at least right here, this is not super condensed DNA. This is euchromatin, which we'll talk more about in a couple of slides. This is readily accessible DNA. You can see these right here. These are the N-terminal end of the amino acid that is folded up to form these histone proteins. And so that amino acid end, or the amine end, where you've got that NH2 on the amino acid, we call this the N-terminus of the protein. This tail is attached to this protein here, making up the octamer. And since we know we have 8 proteins making up this octamer, we should have 8 tails, and we do. we've got 1, 2, 3, 4, 5, 6, 7, 8. One of those tails, or the N-terminus for each of those 8 proteins making up that octamer. There's something very significant about those free tails. It allows other proteins to come in and to modify those histones, which can lead to the DNA unwinding from that octamer. It could also lead to the DNA condensing further. And so you can see some of that involved here. Here is individual histones with the DNA round around it. By the way, the DNA wrapped around a single histone octamer is called a nucleosome. It's an important term to remember. It wasn't part of our key terms, but it is an important one. And so these nucleosomes can then be wound up tighter to form additional condensation. So, and one of the proteins involved in that is histone 1, H1. And so you can see this basically taking 6 of these nucleosomes and wrapping them all into a ring. Now, in every cell in your body, there's about 6.5 feet worth of DNA. And you've got to pack that into the nucleus of that cell, and so it requires a significant amount of condensation. Typically, far more than just nucleosomes, but far less than what you see over here in our super condensed form. So the next step beyond the nucleosomes would be what's called supercoiling. You can see that represented here. Here, each of these is representing a single nucleosome, and then they are packed together into these rings. There's one ring here around H1, and then each of these rings are further packed together in a process called supercoiling. And there are other proteins involved in supercoiling that further. Here we can see the supercoiling that happens in bacterial DNA, these circular chromosomes. And so here is the way we typically represent a bacterial genome or a bacterial chromosome, but in the cell, it would typically take on this form, and you can see that down here. These are electron micrographs of the chromosome inside a bacteria, and typically what we find it is in this super condensed or the supercoiled form. Now, in order for the bacteria to do anything with that DNA, to either replicate it or to write it into RNA, which we'll talk more about in the coming days, it does have to reverse that supercoiling. All right, further condensation, even beyond supercoiling. Over here, we've got prokaryotic DNA, and this is called looping, where the supercoils are wrapped around a central protein. There's a central protein there where you've got lots of supercoils wrapped around that. And then here's each of these loops here that form are made up of thousands or even hundreds of thousands of supercoils, and then those loops can be looped together into bigger loops, and this is basically what it takes to super condense the chromosome down into the form that we see during mitosis and meiosis. And so it is virtually impossible to access any of the genetic information within this structure except at very specific places, like at the site where the two homologous chromosomes attach together during a crossing over event. All right, so this further condensation, unlike the supercoiling happens most of the time to get, in bacteria and especially over here in eukaryotic cells, to pack all that DNA into the nucleus of your cell, pack 6.5 feet worth of DNA. The further condensation typically only happens when the cell is replicating and you don't want access to the genetic material. All right, so heterochromatin, this significantly condensed version. You can see here we have a nucleus. Let's see if I can get my mouse. It's just going to be the pin. So here we've got our nucleus, and in all these regions that are dyed black, that's where you've got chromosomes that are in the form of heterochromatin. A few things about heterochromatin. It is highly condensed regions of the DNA, or you could have entire chromosomes as heterochromatin. In our inheritance crash course, we talked about that female mammals and female Drosophila have 2 X chromosomes versus the 1 X chromosome you find in males. And so the female mammals and female Drosophila, they have to completely inactivate one of those X chromosomes to avoid having a situation of overexpression of the genes on that chromosome. And so sometimes an entire chromosome, like an X chromosome, will be in the form of heterochromatin. Now, this is unique to eukaryotes. Prokaryotic chromosomes do not take on this fully condensed form that is virtually inaccessible. So this is what we call mostly inactive. It is not irreversible, which is not why it is not entirely inactive, but it is energetically expensive to reverse it. Now, euchromatin. Euchromatin is found in prokaryotes and eukaryotes. And here again, we've got a nucleus here. And in all the areas stained black, we've got euchromatin. And and at least in eukaryotes, you have that nucleosome, or what we call beads on a string, colloquially. And so it's not completely decondensed, if you will. It is when it's at least the section that's being replicated or the section that's being copied into RNA, but the rest of it actually stays in this beads on a string with the DNA wrapped around a histone octamer forming this nucleosome. So it still gives us, and then you could even supercoil that, and it still gives you access to it. It's accessible by the machinery that need to replicate DNA or the machinery that need to copy DNA into RNA, but it still shortens the DNA enough that you can pack it into the nucleus. So this is mostly active. And so if you just compare X chromosomes, one X chromosome, the bar body, is going to be heterochromatin and is going to be incredibly condensed. The other X chromosome is going to be mostly euchromatin and is going to be mostly active. Now then portions of individual chromatins may be shut down as heterochromatin, and these are some of the epigenetic features that we were talking about in our crash course on inheritance. And so it's somewhat random which X chromosome gets shut down. It's somewhat random which portions of chromosomes get formed into heterochromatin, but it's highly regulated and highly controlled, even though it may be random based on whether it's a section on the paternal chromosome or a section on the maternal that gets shut down. But as far as specific location, it is very highly regulated. All right, so the centromere. Centromere, again, it's roughly the middle of the chromosome. And what you find inside of the centromere, and again, it's the site where the two sister chromatids are bound together. It's also the site where the microtubules actually physically attach to the chromosomes to move them around during mitosis and meiosis. It's also one of the most accessible sites on the chromosome when it's in its heterochromatin form. And the reason for that is because you have several proteins that bind to the centromere region. And so here you can see this CENPH1 complex, which is made up of 1, 2, 3, 4, 5, 6, 7, 8, 9 different proteins. And that is a site where we've got a lot of protein attachment to facilitate holding the sister chromatids together, or to facilitate the microtubules remaining attached to those chromosomes until the time at which they separate. Now, these proteins recognize a very specific region on the chromosome, and it's not just that they're looking for the halfway point, they're looking for a very specific sequence. And so in these centromeres, we find highly repetitive sequences that these proteins are looking for to determine where the centromere is. And these proteins, we call these syn proteins, centromeric proteins. And that should say bond, not bund, the second typo, bond to the centromere. And again, this functions to hold sister chromatids together, also functions to attach chromosomes to what's called the kinetochore. And we'll see that machinery in a couple of weeks when we talk about cell cycle. All right. Last thing we're going to talk about in preparation for our class on Monday is the end of the chromosomes, called the telomere. Now, the telomere, like the centromere, also has very highly repetitive sequences. Now, for the telomere, that serves a dual focus. In the centromere, it just serves a focus of telling the syn proteins where to bind. In the telomere, it actually serves a dual focus. One is to show an enzyme called telomerase where to bind, and we'll talk more about telomerase in the coming weeks. But telomerase is an enzyme that actually protects the chromosome as it shortens. Every time, we'll talk about this on Wednesday when we talk about DNA replication, but every time a chromosome is replicated, at least a linear chromosome that you find in eukaryotes, every time a chromosome is replicated, it shortens a little bit. And you'll see why that is on Wednesday. But there's an enzyme called telomerase that will come in and will rewrite the part that was lost. And so, again, one part of that highly repetitive region in telomeres, and it's very specific to that region, the repeated units, is so that telomerase knows where to bind, and makes it very simple, actually, for it to just rewrite more of those repeats. The other function of the repeated region in the telomere is for something really important to happen to protect it in between when it's being replicated. And so here you can see this. If this were all stretched out, if we were to take this loop here and stretch it out, the end of the chromosome would be way over here off the screen. But you can see that there's this 3' end right here. I'm following it right here. So this 3' end of this chromosome actually extends all the way through to here. So the 3' end is longer than the 5' end at this telomeric region. Now, the 3' end is where you have these repeated units. You also have those repeated units in this section of the chromosome here, in this light gray, not the blue, but the light gray. And so this free 3' end is actually, it recognizes that, and it comes in and basically outcompetes this region here for that strand. And so what happens is you get this loop that forms, so that this section here that would have been bound to this portion of DNA is outcompeted by this free 3' end, and so that forms this little bubble here called the D loop, and then this whole structure, this whole extra portion of the strand loops around, forming what's called the T loop to go back in, so that that free 3' end can actually go in and bind to that other strand. And so what this functions to do is to protect the end of this strand. So you would normally just have a free 3' end right here sticking out, exposed, and exposed strands of DNA tend to get degraded very quickly. And so as DNA is double-stranded, it protects itself, but free empty strands either get degraded or machinery will come in and write the complementary strand if they can. We'll talk about some challenges with that at the telomeric end. And so looping this around actually protects that free 3' end from both of those. Machinery can't get in to write a complementary strand because it's already bound to a complementary portion, and machinery can't come in that would degrade that and take it down. And then so all of these here are proteins that function to stabilize and support these loops and to ensure that that loop does not disappear until it needs to, until the cell needs to access something at this telomeric end. You don't have genes out here at the telomeric end, but you do need to access it when the cell is copying the DNA so that it can ensure it copies each chromosome correctly and forms daughter cells that are genetically identical to the parent cell. And so this is really interesting. You see the citation here, Nature Reviews. They reviewed a paper that was published in Cell in 1999 that discovered this loop. And the title of the paper, you might get a kick out of this, is Telomeres do a deloop T-loop. And so that's the title of the paper, and in them explaining the presence of this D-loop here and this T-loop here, the deloop T-loop. And so I thought that was pretty neat and worth sharing. But again, both the centromere and the telomere have these highly repetitive regions that are either, or actually in both cases, recognized by proteins, and the centromere proteins to bind to those areas, and the telomere proteins to actually write one of the strands longer, and then also in the telomeric region to facilitate that free 3' end getting in and binding to that complementary portion. So that's it for this lecture. As you prepare for our class on Monday, please remember to take quiz 2, which is posted on Canvas. And please also be prepared to have some discussion on these topics.